Hi students, welcome to the biochemistry class. Now let us go into see the topic is metabolism of amino acid, phenylalanine and tyrosine. This is the structure. You can see the picture here. This is the structure of the phenylalanine and tyrosine. Phenylalanine and tyrosine are mostly related aromatic amino acids. So what are the contents we have to learn here? The first one is the conversion of the phenylalanine to tyrosine. That means the phenylalanine amino acid, how it is converted into tyrosine. Okay, and the second step, degradation of the tyrosine. That means the metabolism of the tyrosine activity. And the third one is the synthesis of the melanin. How the melanin, okay, melanin is one of the pigment. How the melanin pigment is formed. Okay, that is the synthesis. And the fourth one, synthesis of the thyroid hormones. Some thyroid hormones are synthesized. Okay, that is the fourth step. And the fifth step, biosynthesis of the catecholamines. And the sixth step, disorders of the tyrosine metabolism. So, due to the deficiency of the or due to the metabolic activity of the tyrosines, what are the disorders? Okay, we can see one by one. The first one, the conversion of the phenylalanine to the tyrosine. How the phenylalanine, it is converted into the tyrosine. This is the first step. Okay, so phenylalanine, it is converted to produce the tyrosine by the enzyme, that is the phenylalanine hydroxylase. Okay, in the presence of the enzyme, the name of the enzyme is called as phenylalanine hydroxylase. Okay, phenylalanine, it is converted into the tyrosine. Okay, so it requires the enzyme that is known as biopterin. Usually the active form of the biopterin is tetrahydrobiopterin. Okay, so in the phenylalanine hydroxylase reaction, in this reaction, what is the first step? That is tetrahydrobiopterin. It is oxidized into dihydrobiopterin. That means 4H4. It is converted into H2. Okay, the tetrahydrobiopterin is then again it is regenerated by the NADPH dependent dihydrobiopterin reductase. Okay, with the help of the NADPH dependent dihydrobiopterin reductase. Okay, it is again regenerated. Okay, you can see the picture. Through this picture you can clearly understand, okay, the phenylalanine, how the phenylalanine is converted into the tyrosine. Okay, so here phenylalanine hydroxylase, in the presence of the phenylalanine hydroxylase, then H2 biopterin, that means dihydro, H2 means 2. Okay, dihydrobiopterin is converted into tetrahydrobiopterin, isn't it? Okay, in the presence of dihydropteridine reductase. Okay, with the help of the dihydropteridine reductase enzyme, the di is converted into the tetra. Okay, then again it is repeated again. The hydroxylase enzyme, it is repeated. Okay, it is the formation of the tyrosine molecule is formed from the phenylalanine molecule. Okay, usually the name of the enzyme, that is phenylalanine hydroxylase enzyme, isn't it? Okay, this is the phenylalanine hydroxylase enzyme. Okay, it is involved mostly, but it is present in the liver. Okay, so when there is a defect in the phenylalanine hydroxylase, when there is a defect in this enzyme, means the conversion is blocked. Okay, so this conversion is blocked. Okay, when there is a defect, when the phenylalanine hydroxylase, this enzyme is defect in the liver, means the conversion is affected. Usually the phenylalanine is converted to the tyrosine with the help of the phenylalanine hydroxylase enzyme, isn't it? Okay, when it is blocked, okay, when it is defect, mean this reaction is blocked. Okay, so what is the result of the what is the result means the disorder of the phenylketonuria. Okay, so when the conversion is affected, conversion of the phenylalanine is converted into the tyrosine is affected, means it will cause the disease, that is the phenylketonuria. Okay, and the second one, degradation of the tyrosine. Okay, so metabolism of the tyrosine. These are various steps are available here. The first one, tyrosine, it undergoes the transamination. Okay, the amino groups are transferred here to give the hydroxyphenyl pyruvate. Okay, the first step, this is the first step. Okay, each and every step we know in the biochemistry, every step is catalyzed by the particular enzyme, isn't it? In the presence of the enzyme only the reactions will be carried out. Okay, so here also one of what are the enzymes are involved here. Okay, step by step we can see. The first one is the enzyme is catalyzed by the reaction is catalyzed by tyrosine transaminase. Okay, so this reaction is catalyzed by the tyrosine transaminase. Okay, and the second enzyme hydroxyphenyl pyruvate hydroxylase. Okay, this is the second enzyme, or otherwise called as dioxygenase. Okay, it is also called as the copper containing enzyme. So what is the role of the enzyme? Okay, it catalyzes oxidative decarboxylation and hydroxylation. Okay, it is involved in the two process. One is the decarboxylation, another one is the hydroxylation. Okay, so such enzyme, okay, what is the role of the enzyme? It converts the hydroxyphenyl pyruvate into homogentisate. Okay, hydroxyphenyl pyruvate is converted into the homogentisate in the presence of the dioxygenase enzyme. Okay, and the next step, homogentisate. Okay, it is converted into the another format. Okay, so what is the role of the another enzyme? That is homogentisate oxidase. Okay, so such enzyme, it break down the benzene ring of the homogentisate. Okay, then only it will develop into the 4-mallyl acetoacetate. Okay, so the enzyme homogentisate oxidase, it is converted the homogentisate into 4-mallyl acetoacetate. Okay, then another next step, another reaction. Now the mallyl acetoacetate, it undergoes the isomerization reaction. Okay, it will develop into the isomers. Okay, that means it will develop into the 4-fumaryl acetoacetate. Okay, mallyl acetoacetate is converted into the 4-fumaryl acetate. Okay, what is the role of what is the enzyme is involved here? That is mallyl acetoacetate isomerase enzyme. Okay, mallyl acetoacetate isomerase enzyme is involved here. Okay, then only mallyl acetoacetate is converted into 4-fumaryl acetoacetate. Okay, so what is the next following reaction? Okay, fumaryl acetoacetate. Okay, so this is the enzyme. In the red color, I have marked here as a enzyme name. Okay, so this is the another enzyme, fumaryl acetoacetate. Okay, so now what is the role of the enzyme here? Okay, so it brings about the hydrolysis of fumaryl acetoacetate is converted into fumarate and acetoacetate. Okay, so the fumaryl acetoacetate is converted into fumarate and acetoacetate in the presence of fumaryl acetoacetate enzyme. Okay, so finally the fumarate, the final product is the final reaction is the fumarate. Okay, is formed. It is the already we have studied in the citric acid cycle, isn't it? Okay, so fumaric acid, malic acid. Okay, the formation of fumaric acid, malic acid we have studied in the citric acid cycle, isn't it? So fumarate is an intermediate of the citric acid cycle, and also it is a precursor of gluconeogenesis. Okay, we have studied in the carbohydrate metabolism what is glycolysis, gluconeogenesis, glucogenesis, isn't it? Okay, this is the form precursors. Okay, the initial product, initial formation. Okay, then this is the flow chart of the tyrosine degradation pathway. Okay, so the basic step, how the phenylalanine is converted into the tyrosine. Okay, in the presence of the phenylalanine hydroxylase. Okay, this is the first step we have discussed earlier. Okay, then the tyrosine, how the tyrosine is converted into the 4-hydroxyphenyl pyruvic acid in the presence of tyrosine aminotransferase enzyme. Okay, the next one, 4-hydroxyphenyl pyruvic acid is converted into homogentisate. Okay, in the presence of 4-hydroxyphenyl pyruvic acid dioxygenase, isn't it? Okay, then again homogentisate, it is converted into the mallyl acetoacetic acid in the presence of the homogentisate 1,2-dioxygenase. Okay, again the mallyl acetoacetic acid is converted into fumaryl acetoacetic acid. Now the fumaryl acetoacetic acid is converted into fumaric acid, and also fumaric acid is combined with the acetoacetic acid in the presence of fumaryl in the presence of fumaryl acetoacetic acid hydrolase. Okay, this is the formation of the fumaric acid and acetoacetic acid. This is the tyrosine dehydration pathway, second step. And the third step is the synthesis of the melanin. Okay, first let us see about what is melanin. Okay, we should know about the meaning of the melanin. Melanin means it is the pigment, isn't it? Okay, the pigment of the skin, hair and the eye. Okay, so tyrosine is the precursor of the melanin. Okay, so the melanin is developed or formed from the tyrosine only. Okay, so the name of the enzyme is involved here. What is the main role of the enzyme here? That is the tyrosinase. It is a copper containing oxygenase enzyme. Okay, with the help of the enzyme only, tyrosine is converted into the melanin. Okay, so this is the step here. Okay, okay, the step here, that is tyrosinase hydroxylates tyrosine. Okay, it is developed into the DOPA. DOPA means that is 3,4-dihydroxyphenylalanine. Okay, the next reaction, tyrosinase. Now DOPA is converted into dopaquinone. Dopaquinone is converted into leukodopachrome. Leukodopachrome is converted into 5,6-dihydroxyindole. And again the 5,6-dihydroxyindole is converted into indole 5,6-quinone. Okay, so with the help of the enzyme tyrosinase. Okay, so and another one, the last one, the quinone, it is developed into the melanochromes. Okay, melanochromes are formed from the indole quinone. Okay, then again finally converted into the black melanin. Okay, this is the biosynthesis of the melanin. Okay, so tyrosine, then dopa, dopaquinone, then leukodopachrome, then 5,6-dihydroxyindole, then indole 5,6-quinone, then finally melanochrome is developed. From the melanochrome, melanin black polymers is developed. Okay, this is the synthesis of the melanin. This is the flow chart explaining about the synthesis of the melanin from the tyrosine.